DNA is made up of a phosphate and sugar backbone that attaches to nitrogenous bases. When they are all connected, that is called a nucleotide. These nucleotides make up the double helix shape, which is like a twisted ladder. The discoverers of this structure were Wilson and Crick. They discovered it through a photo taken of DNA. Complementary base pairs are pairs of nitrogenous bases that only connect to each other, such as adenine and thymine, or uracil and mRNA, or guanine and cytosine. Adenine and guanine are purines, which means that they have 2 nitrogen rings. Thymine or uracil and cytosine are pyrimidines, which means they have 1 nitrogen ring. Here you'll see DNA splitting into 2 strands so that the mRNA can start to be formed. mRNA is what is used to make proteins during translation. Transcription is the creation of mRNA. The structure of mRNA is similar to DNA, except instead of thymine, you'll have uracil. And through this process, a purine and a pyrimidine must pair up together. mRNA will use one of the strands of DNA to template itself off of. With any T that is on a DNA strand, an A will form. With any C, a G will form. With any A, a U will form. And with any C, a G will form. This all happens in the nucleus of a cell. The mRNA can then leave the nucleus to create proteins, to start the process of translation. After transcription is done and mRNA is made, it will then go to the ribosome in order to make proteins.
The ribosome gives the mRNA and tRNA a spot to create proteins. Translation is the creation of the protein, where the tRNA brings amino acids based on a code that is read on the mRNA. tRNA is the transport RNA, which will move the amino acids connected to anticodons. Codons and anticodons are basically like complementary pairs of each other. In a codon, say it is AUG. The anticodon for that would then be UAC. And based on the codons in the mRNA, it will then begin constructing proteins. These pair together, the tRNA checks using the anticodon to make sure that it's the correct amino acid. The amino acid chain is then finished once a stop codon is inscripted on the mRNA, and the protein is then let go. This is a codon chart. Codon charts are used to tell which amino acids match with which codons read from mRNA. You can tell which amino acid matches with which codon by reading these charts. You would start off in the first column of letters, U, A, C, and G. The first letter in the codon tells you which row to look at. Then in that row, you find a second letter, U, A, C, or G. That then tells you the column that you go down. This will give you a square in the 4x4 grid. Within that square, there are 4 lines. Again, you find the third letter in the codon, U, A, C, or G. This will tell you which row within that square to look at, which will tell you which amino acid matches with that mRNA codon.